Hello, Dr. Ganguly and Faljuna Mohra. So, we're happy to have you in this PDB webinar. And so, we are like a volunteer organization from RSG-India. I think we will give intro about our organization in the upcoming slides. So, we will take 5 minutes for our introduction for RSG-India, and then we will start our session with your talk. And we will wait for another 5 more minutes, and we will start at sharp 3 o'clock. Yeah. In the meantime, Akriti and Nikita, if you want to add anything here, you can.
So yeah, welcome, Dr. Ganguly.
Researchers in India. And yes, we are really looking forward to feedback, questions. We'd be happy to stick around for a bit and try to help people as much as possible.
Yes, for this session, we have lots of fresh minds, and they have lots of questions in the, with them, and definitely we will have, I think, lots of time for the question and answers. So as you can see, so and for this work, for this session, we have participants from all the spectrum. As the Akriti also mentioned, that we have participants from 7 different countries as well, not only from India. We have participation from 7 different countries, and with some of them are joined from the industry, and some of them are from postdoctoral and assistant professors. And I think everyone is excited for your talk. And we will wait for another 2 more minutes, and yeah, we'll start at 3. A small request to all the participants: if you have any questions, you can chat in the in the chat box. So after the talk, we can have question and answer session. Okay, we will start the session now. So, welcome everyone. Welcome to RSG-India's Meet and Learn with the Bioinformatician series. This is episode 2 of MLTB series, and in this episode, we are going through a journey through the protein data bank and focus on PDBE resources. We have two bioinformaticians from PDBE, and without much wasting time, I would like to welcome Nikita to give introduction about RSG India. So who we are and what we do through our organization. And I welcome Nikita to give intro about RSG India.
Thank you, Govind. I think I'm audible to everyone.
Yes,
So,
Nikita, you are. Yeah.
thank you, ma'am, for this accepting our invitation and providing us opportunity to being a bridge between the young learners and the expertise, so that they can explore more and can enhance their skills. So, by not wasting too much of your time, I would just like to introduce RSG India a little bit, because this has been a platform that people can explore for their skill development and to learn a little bit more about bioinformatics and computational biology. So, RSG India is not just a single organization. It is a sub organization. Like, it is a student and volunteered organization. So it is basically parented by ISCB, which is International Society of Computational Biology. It was a US based organization which aims to just connect with all the computational biologists, and so that they can interact with each other and enhance their skills. So, in order to reach as much as audience possible, they have come up with a student council, because students are the young minds. So they have come up with a student council which is called ISCB-SC, which was established in 2004. So it aims to connect all the students from different backgrounds, like different hierarchy, bachelors, undergraduates, postgraduates, and postdocs as well. So in order to just reach as much country as possible, they have come up with small regional centers or small regional groups. Like, one of us is RSG India. So currently there are 26 regional groups running in different countries across the globe. So one of them is RSG India, as you can see. So RSG India was established in 2006, approximately 2 years after ISCB-SC has been formed. So RSG India is being working since 2006. So I would request go into this in the slides. Yeah. So a brief history of RSG India that I would like to introduce to you is, like, we started in 2006, but some of the years we took to just enhance ourselves. So 2007 to 2008 has been like, we are extracting diamonds from the minerals. Like, we are extracting as much as possible. So to provide information, or being a bridge between the possible solutions or possible opportunities for different students. So after 2018, if you want to look at our work or the organization, you can go to RSG India website. So we have tried to document as much as possible in our website. So some of the members we have joined 2023, and from that we are trying to build as much as possible. So in this year we have conducted 2 webinars currently. Like, one is Academy to Industry for computational biology's tricks and tricks advices that people can use, and then we have another webinar where we learned about science communication, which is very important for any of the scientists working all over, like, in different aspects. So we have come up with something through Brinkadas Gupta. Then we have one of the MLTB series. Like, this is the second series. So we had first series by bioinformaticians from Glycans. So we had it by Raza Mujumdar and Michael Timer. So this is going to be our second MLTB seminar. So if we look at the history, look at the previous year, we had 6 webinars, 5 seminars, 2 workshops, and 1 biohackathon. And currently we aim at multiple countless learnings. in this year. So with this aim, have our core committee, so which includes Govinda Rao as the president, Nikita, which is me, as the secretary, Akriti as communication advisor. She was our previous president for RSG-India in the last year. Then we have Ashita as nodal head, Sabyasachi as design head, and to have interaction and faculty as advisor, we have our faculty advisor, Dr. Manish Kumar, who is helping us with taking decisions and bringing better solutions. So in this, with this, I would just say this is the second series of MLTB, and this is going to be. I wish this is going to be a very important thing for a student. So if we have a glimpse of 2023, we had 7 episodes of MTB series, and over 500 participants have participated in MTB series. MTB series basically a series which is similar to MLTB, but MTB just had we discussed with bioinformatician, but here we are aiming in MLTB to learn from bioinformaticians. So here we would like to show you a glimpse of the MTB series in the previous year. So we had 7 episodes with in multiple months. So you can see Dr. Jairam and Dr. Alok Bhattacharya and so many renowned scientists who came up in these MTB series, and we had a lot of learnings. Hope this learning is continued in MLTB as well. So thank you.
Yeah, thank you, Nikita. Thank you for introducing RSG-India. And I would like to welcome Jatin to introduce our today's speakers. And over to you, Jatin.
Thank you. Thank you, RSG-India, for giving me the opportunity to introduce Dr. Sudakshina Ramani. Sudakshina completed her PhD from Bose Institute, India, where she was interested in determining the structures and biophysical characterization of nucleic acid and ligand complexes. Thereafter, she did her postdoctoral studies at Cancer University, USA, where her research were focused on interactions of RNA with aminoglycoside antibiotics. Sudakshina joined the PDP Europe team in 2023 as a scientific database curator, where her work involves annotation and curation of preliminary PDB and EMDB submissions and extraction of relevant biological information. She is also a part of the PDP Europe training and outreach team, where she is actively engaged in developing training material and outreach activities such as delivering workshops, webinars, etc., which are aimed at researchers to leverage existing data in the PDB. So next, I have the honor to introduce our second distinguished bioinformatician, Dr. Farina Mamania. Dr. Farina Mamania is a bioinformatician at the PDB in Europe, where she focuses on broadening the understanding and use of alpha-4. She completed her PhD at the University of Nottingham's School of Pharmacy, specializing in the role and expression of SLC transporters in lung epithelia within the division of advanced drug delivery and tissue engineering. Her doctoral research provided critical insights into the structure and function of these proteins, utilizing CRISPR-Cas9 for gene editing and transcriptome analysis. At PDB Europe, Farina works on making the AlphaFold database accessible to everyone, developing comprehensive practical guides, and conducting outreach on AlphaFold applications. Additionally, she designs and delivers training programs to enhance the understanding and utilization of AlphaFold in the scientific community. So today, Dr. Ganguli and Dr. Mamania will share their insights on a journey through the protein data bank, which focuses on the PDB Europe resources. So we welcome you both in the MLTB episode 2 of RSG-India. Now over to you, Gopal.
Yeah. Thank you.
Thank you. Sorry.
Yeah.
Please go.
Yeah, sorry, ma'am. Yeah, thank you, Jatin, for introducing our speakers. And you can see the responses for our webinar for today's event. So we have broad spectrum of audience from for this episode, and so these are some pre-webinar surveys, and you can see how the people are using the PDB resources. And yeah, I would like to hand over it to Dr. Sudakshina and Paulina Magna. So the stage is yours.
Thank you. I'll share my screen.
Yeah, sure.
Please let me know if you can see it. Hiya. Can you see the screen now?
Yes, ma'am, we can see the screen. Yeah.
And you can hear me all right?
Yeah. Everything is all right now. Fine. Yeah.
Great. Okay. So I am a structural biologist by training. I will try to take you on a journey through the protein data bank, and then my colleague Paulina, she'll go through the AlphaFold database, which is another part of the PDB. I'm going to keep my camera off because I tend to move my hands around a lot. I don't want you to see that, but I'll turn it off at the end. And so as I think Govinda mentioned, please keep your questions for the very end. We'd be happy to stick around and discuss all your questions. Okay, so if you have no idea what the protein data bank is, we are an archive of experimentally determined 3D structures of biological macromolecules, which means we have proteins, nucleic acids, and any small molecule or ligands bound to these macromolecules in our database. You may be familiar with some of these landing pages. So the WWPDB, or the Worldwide Protein Data Bank, has many different partners. RCSB-PDB and BMRB, which is the Biomagnetic Molecular Resonance Data Bank for NMR data. They are based in North America. In Europe, we have PDBE, Protein Data Bank in Europe, and EMDB, which is the Electron Microscopy Data Bank. We are based in EBI in Kingston. And in Asia, we have PDB Japan, based in Osaka. So all of these different partners, they come together and form what we call the WWPDB. But each of us have our own pages and own websites. So if you've never seen what the Protein Data Bank in Europe page looks like, this is our landing page. We have a search bar right at the top, but we also provide a number of different services. You may have used some of these services according to the survey. So we have PDBE PISA, which is used to predict macromolecular interfaces, PDB Fold for alignment of protein sequences, and PDBE Chem, which is a dictionary for small molecules. We also provide the archive statistics. So as of this week, there are more than 220,000 structures in the PDB database. Now, what exactly is a PDB code that you may be familiar with? So whenever you deposit to the protein data bank, you are given a code. Generally, it's a 4-character alphanumeric code. And this has to be referenced in any publication that you have. Now, however, all the entries in our database may not have a publication corresponding to it. Some of these may yet be unpublished. You may also have seen these validation statistics, these sliders that we have for each and every entry in the database, where the color red signifies worse and the color blue signifies better. And there are a number of different metrics that we show. And depending on the values of these metrics, you can actually determine how good or how not so good the modeling for that particular entry is. I believe you're mostly computational biologists, so you would know what a PDB file format looks like. In case you don't, it's just a text file. It has fixed column width, and it has very preliminary information such as the chain name, residue number, etc. We are slowly phasing out the PDB format. And now it's the MMCIF format, which is the master format that we are using for the database. It's still a text file, but it's much more computer readable. It's extensible and verifiable. So that means you have key value pairs for each category. And it's also loopable, which means that if there are multiple values for one particular category, you can still have them in the file. If you have never downloaded and seen what a CIF file looks like, please go ahead and do that. There's a very succinct user guide available now that we have published very recently. Please go through it. We use particular MMCIF parsers for parsing our files, which is again present in GitHub. And if you want to talk to us more about this file format, we'd be very happy to discuss this. The growth of data in the PDB has been exponential over the years. We reached the milestone of 200,000 structures in early 2023. And as I showed you this year, this week, we have more than 220,000 structures, which means there's a lot of data out there. How do you cut through all the noise and get something that you are really looking for in our database? So how exactly do you search for structures at PDBE? I previously shown you the search tab. Whenever you start typing something in the search tab, you get a lot of hits, a lot of different categories. You may be looking for enzymes, anything with a gene ontology mapping. If there's any ligand that has the term in it, they will all come up as search terms. In this particular case, I'm looking for hydrolases. So I will click on the enzyme hydrolase, and this is what I see. This is what we call the entry, the search page. And as you can see, there are more than 21,000 structures for hydrolases in the database. So how do you refine the search? How do you make it more suited to what you are looking for? You use these filters or facets, as we call them, on the left-hand side of the page. Suppose you want to look for one particular organism, such as human hydrolase. You can select this, and now they will be present in the search bar. So as you can see now, the total number of entries are just over 6,000. So you've really cut down the number of entries. If you want to further refine your search, there's the option for advanced search available. As soon as you click on it, you get this custom form where you can customize the different search fields depending on what experiment type, what molecule type, if you're looking for ligands, etc., etc. Now, by default, if you've never seen this before, the search page always opens on the entries page. You are also able to sort by the date. Suppose you want to search from latest to oldest, or even depending on the resolution, you want to have higher resolution first, you could be able to sort by them. Now, what do these different tabs mean? Suppose if you go from the entries to the macromolecules tab right over here, this is showing you how many unique macromolecules are present. So there were 6,000 entries, but not all of them were unique proteins. This is telling you how many unique human hydrolases are present. So that's just over 2,000. For example, the first one, which is this particular hydrolase, is present in 3 other entries, which is being shown right over here. So this is again helping you group the data, refine your search, and really understand what you're looking for. The next is the compounds tab, where if you have any ligand or small molecule bound to a particular structure, a particular protein, this is where it will come up. If you're looking for a particular ligand, you can also search for it. You get all the protein name, the ligand names listed right over here. And finally, you have the protein families tab, where they are grouping all these different macromolecules into particular protein families and showing you. So the aim of me telling you all of this is so that you can get more information out of the search results. Now, okay, you have your structures, right? You have a list of your structures. You need to see what they look like. You need to visualize this. And what we use for visualization is a software called Moldstar. I know many of you are familiar with PyMol or Chimera. If you've never used Moldstar, I would really ask you to go to our website and play around with it, and I'll tell you how you can access it. So it's the good thing or interesting thing about Moldstar is that it's an online structure viewer, so you do not need to install it. You can use it online. And there are lots of additional features that, and I'll show you some of them, which are really, really interesting. So how do you get to MoldStar? Suppose if you have an entry that you're looking at, you have this tab available right here, which is called the 3D Visualization tab. As soon as you click on it, this is the view that you see. So this is the MoldStar view. You can click upon it, you can play around with it, drag it. The sequence is present right over here, and there are additional tabs that you can use to view in different modes on the right. You can also access it from the PDBE search page. So if you have a list of entries, you have this tab right here, 3D visualization, and it will lead you to the same MoldStar view that I have already shown you. Now, suppose if you're viewing a crystal structure, say, and you want to see the electron density, you can turn it on using MoldStar and the map and then display. So this is actually showing you how well the structure, the coordinates have been modeled, and with respect to the actual experimental information, which is the electron density. If there's positive or negative density, they will also be highlighted in different colors. So you know if there are missing atoms or if something has been overfit into the density. This really gives, even if it's a cryo-EM structure, you will also have a map to support your experimental data, so you can visualize all of that using MoldStar. Another very interesting feature is you have this tab called validation, where you can actually highlight the different issues on your structure using the software, which means if you pull down, if there are any side chain outliers or if there are any steric clashes in the structure, you can play around with this tab and it will actually be highlighted where exactly these clashes are, where the outliers are, which part has been modeled with greater confidence compared to a different part. You get much more comprehensive understanding of the structure just from the 3D view. And finally, if you click on this arrow right over here, it will give you the option to select residues versus atoms versus chains. You can also keep on adding different views to the components so that you can view them one after the another. So if you're preparing, say, a presentation and you need a high-resolution video or a high-resolution image, it's really easy to take a screenshot, to download it, to prepare custom videos using different state snapshots. It's really easy to use. Please go ahead and try to play around with it. Let us know your feedback. We're always very happy to get feedback from our users. So yes, let's move on then. Sorry if I'm moving too fast, but I have a lot to cover. Any questions are welcome at the very end. So now you have a list of structures. You have, you know how to visualize these structures, but what else is the page telling you? What other information can you get just from looking at an entry? If you move down the page, you have this particular tab, which is called the structure analysis, and it gives you information about the length, the weight, UniProt ID, any sequence domains if it's annotated. You can click on this molecule details view. It'll take you to what we call the 2D prot vista viewer right over here. This is highly interactive in the sense that it'll give you a view of the chains, domains, secondary structure. All you need to do is hover your mouse over it, and it will tell you all the information. Suppose if you want to know more information about, if you want to know more information about the domains, you need to hover your mouse and it will show you exactly which helix that is. There's a 2D view right over here, which will tell you which part of the helix you're looking at, which helix you're looking at, and that is the one that also gets highlighted in the 3D structure on the right. So it's you can relate the 2D structure with the 3D structure in the actual entry. There are a lot more information available. I am not able to go into details. You have information about ligand binding sites, interaction surfaces. Maybe we can have future webinars that really focus on how you can utilize these. Now, suppose if you are interested in looking at small molecules, ligands that are bound to proteins. On the entry page, you have a tab called the ligands and environments right over here, which will list all the different ligands that are present. Suppose I want to know more about this particular ligand. All I have to do is click right on the ligand name, and it will take me to this view, which is a very interactive view again. So this is actually showing you the ligand in 2D with all the amino acids that are close to it, and in 3D, where the ligand is actually present in the ligand binding pocket. So if you hover your mouse over any one of these entries, any one of these residues, suppose for this one, it will highlight this particular amino acid in the 3D figure. So you can actually see where it's positioned with respect to your ligand, how it's interacting. And you may have seen they all have different colors. So these colors have particular meaning. So they are all, if you go through to these 3 dashes, they'll give you more information about what each of these different colors mean, what they symbolize. So you know which residue is interacting how with the ligand, you get a 3D view. So you are getting, without even downloading the structures and having a different molecular viewer to visualize, you get all of this information just from the entry page. Now, because I want to focus more on other tools that we have, I have listed a number of tutorials and trainings and webinars that we have on searching the PDB for information. Please go through them. We also have API webinars about how you can programmatically access information from the PDB. Please go through them. And yep, how am I doing on time? Am I good? Great. Okay, so moving on. I am going to tell you something very interesting. And I think from the pre-webinar survey, I think most people have not used this service before. So I really want to focus on this. It's called PDB-KB, or the knowledge base, aggregated view of proteins. So just looking at a 3D structure, what do you know? What do you get to know? What information do you know except how it looks, how the protein looks? Basically, nothing. Just 3D coordinates by themselves do not give you any relevant biological information. What you need to do is you have to get the structural context. For example, if it's a homodimer, if it's a tetramer, because only when you know the structural context do you also know the biological context. For example, the protein that I'm highlighting is the SARS-CoV-2 main protease, and it has to be in a homodimeric condition to actually function for the catalytic dyad. You need to know where the ligand binds to actually get maybe an inhibitor or a drug which can target this particular protein. So what is important is looking at proteins or nucleic acids in the context of its entire biological function, not just the 3D structure. So you need extra value-added annotations to give actual meaning to your coordinates. If you go out there, if you go to the internet, there are so many different websites, different databases that provide specific annotations. I've listed just a few of these. So you have PFAM and you have RFAM, which give you information about protein and RNA families. You have Interpro for protein domains. You have, say, metal PDB, which gives you information about metals in the structure. But they all provide different annotations, and we did not previously have one place where you could access all of this information. So we had the PDB entries. There were specialist databases available which provided this information. And now we have interconnected and annotated these PDB entries with all of this information about domains, about ligand binding sites, drug binding pockets. All the extra annotation are now present in our KB pages. So how do you access our KB pages? Before I go into the details, you can just go to Google and type in PDB-KB. It'll take you to the page. If you are on our entry pages, you have a link right over here. These are the UniProt IDs. So this is basically the protein that is linked to this particular UniProt ID. And you can also access it from the UniProt entry pages as well. And this is what the KB page, the landing page, looks like. This is what we call the summary view. And the summary view, it gives you information about the protein of interest. If you have a particular gene that is, or a protein that has an enzyme number associated with it, you can visualize that enzyme number. Also, if there's a disease that is associated with it, that will be visible. So all you have to do is hover your mouse over it, and it will give you all that information. There are a number of icons below the relevant page to take you, and these are basically the tabs, but you also have the shortcuts, the icons right over here. And I'm going to go through each of these pages in details. Let's start. Before we start, so we also have the 3D Moldstar view showing you the PDB chain covering various segments. And all you need to do is hover over this tooltip to really know what the color schemes mean, what you're looking at. All of that information is present. So when we come to the structures tab, suppose I have one particular protein that I'm interested in. What this tab is telling you at first glance is there are 890 instances of this particular protein with that particular UniProt code in the PDB database. Now, this is again the Prot Vista view, which I've shown you previously. This is listing all of the different PDB entries that has this particular protein present. By default, the first protein is the one, so this one. This is the protein that is being highlighted in the Moldstar view. If you click on the next one, that would be the one that is being highlighted, and you can scroll through each of these. Yeah, this is what I just said. The first one is selected by default. Now, you can click on any PDB segment to basically display the structure in Moldstar. So basically, if you hover over it, if you click on this segment, this is the one that gets focused on Moldstar. This is the one that is being highlighted. Now, remember that all of these that have been listed are actually experimentally determined structures. And you see these tiny icons right next to it? They have specific meanings too. So if the green symbol is on for the first one, it means it's present. The protein is present as part of a complex. If the second one shows whether any nucleic acid is present in the structure, and the final one is showing that, yes, there's a ligand present in this particular entry. And of course, the resolution, if it's a crystal or cryo-EM structure. So basically, just at a single glance, you will be able to know if it's a complex, if there are ligands present. So you can also filter out stuff if you're not interested in looking at structures with ligands, for example. You get information about the structural variation, which part is the helix, which is the part is the loop, which part is the strand. You also have information about domains. These are integrated information, and all of this annotation is coming from different databases like PFAM, CAT, and SCOP. And finally, you also have a tab called Other Structures, where if you have any predicted models from, say, AlphaFold, Swiss model, these are also present. So you have a complete overview of the protein, of all the different structures for that particular protein that you're interested in. The Ligands tab is also interesting. So the Ligands tab is telling you if there are 890 entries in the PDB where this particular protein is present, there are 55 unique instances of ligands that are bound to this protein. So it's important to note that only those ligands which are directly interacting with the protein are highlighted. But if you click on this box, all the proteins, so some of these may actually be crystallization agents like metal ions or PEG or some other lipids, they will all be shown if you click on this box, but by default, they're not shown. If you have a particular ligand that you're interested in, you can filter by the name. Also, if there's any known annotation for the ligand, for example, if it's a cofactor-like ligand, if it's drug, if it's peptide-like, they will all be highlighted in green. You just need to mouse over the name to get the full molecule name. You can use the download button to actually download this information. There's a link to go back to PDB eSearch if you want to look at other entries in the PDB where this particular ligand is present. Also, there's the option to view in 3D, which means that the ligand is shown in its actual bound state with this particular protein. And if there are multiple instances, as you can see, there are all the different PDBs where this particular ligand is interacting with this protein. You can actually use the drop-down to navigate between the different instances. If you go down the ligand page, you again have a Prot Vista viewer, which shows you which amino acids, specifically which amino acids for this particular protein are interacting with the ligands of interest, with this particular ligand. You need to mouse over it. I'm sure you understand that at this point. And finally, I really like this view. So if you collapse everything, you get this view. So this is actually giving you an idea about which amino acids or which range of residues within the protein are most likely binding to the ligand. So you get an overall view of which would be an important ligand binding region for this particular protein. Moving on, you also have a tab for macromolecular interactions. So this tab is telling you if there are other proteins that are interacting with the protein that you are looking at. So if there are other macromolecular partners in any other PDB entry, unique macromolecules, they are being listed right over here. So there are 17 other unique proteins that interact with your protein of interest. Again, you can filter by name. If there are any annotations present for these interacting partner proteins, they will be present right over here, highlighted. You need to hover over the name to get the full name. You can download the information using the download button. And you can go back to the PDBE page to all those entries which has this interacting partner present. Again, there's a 3D view where, again, you can use, you can look at all the different structures where your protein of interest interacts with unique proteins, how they interact, which are the regions, etc. You get the prod vista view as well for the residues which are involved in the macromolecular interaction interface right over here. Details of the residues, again, will be highlighted. So we have tried to make this as user-friendly, as easy to use as possible. The collapse view is available in this case too, which will give you an overview of the common interaction interfaces with macromolecules. Now that you know how to visualize proteins with ligands and proteins with other proteins, we have recently introduced what we call the aggregated view, we call it the structure superposition. And this is where you can access it. So you have something called the 3D view of superposed structures. And if you click on it, it will open a Molestar view that I'm going to explain in greater details. So what this is doing is showing you the superposed view of your protein of interest and all the different PDB IDs that it's present. So just by looking at it, you know which are the more flexible loops, or where one particular entry is modeled differently to the rest of it. You can visualize where it is really. So remember that you actually need to turn on the visualization. So by default, they're all turned off. So as you go ahead and keep turning these on, more of the structures could be superimposed. Another interesting, very important thing is that if you have any predicted structures from AlphaFold, you can superpose those as well onto this view. So if you can actually compare that compared to your experimentally determined structures, how is it that the predicted models differ? Which regions do the predicted models differ? You will be able to ascertain that. Also, the PAE score, Paulina will, I think, go into details about this later. That will also be displayed on this particular screen to give you greater understanding. So we have also tried to do the same for superposed ligands. We're showing you the so just by looking at it, you know exactly where the ligands bind. So I can say that this probably is the primary ligand binding site right over here. There are also secondary ligand binding sites present right over here and here. But again, you can select, you can toggle which particular ligand you want to look at. If you're interested in carbohydrates, or if you're not interested in lipids, you can toggle these on and off to really, really filter your search and look at what you're interested in. We have two different tabs for ligands and carbohydrates that you should know. Again, please go to our website, go to the database and sort of play around with this to really understand more. If you have any question, any feedback, we would be very interested to know more about it. Okay, so I have to move on. And the next tab is the annotations tab. So what this tab is showing you is we have a lot of different partner resources that have extra annotations available, and we have incorporated all of these extra annotations right over here. So suppose if you have information about metal binding sites or predicted PTM sites, any biophysical parameters, you can come to this tab for your particular protein of interest, and you get all of this information right over here. So if you want to know where we are getting this information, you need to hover your mouse. We'll give you details about the exact partner resource that we are extracting this data from. Residue level details are present in the tooltip. Hover over it and you get them. You get domain mappings, ligand binding sites, interaction interfaces. They will allow you to compare your structure. And again, you have residue level conservation score from UniProt KB. So if you want to have multiple sequence alignment, you can do it right here. You can also download this MSA file. You can do it for 150 residues or less. But yeah, you can have an MSA. You have different amino acid properties, which is really, really important information. Finally, we also have a tab for variants. So if there are any known mutations that can cause, say, disease, or that may be predicted to be causing a disease, you would have different, all of this information present under this tab. So as you can see, the different mutations, they have different colors responding to different zone information. And yeah, if you're someone who's interested in looking at variants, protein mutations, this would be a really useful tab for you. Second last one is the similarity tab. So what this tab is telling you is how many proteins with more than 90% sequence identity to your protein of interest are present. Important to note, there are two tabs right over here. The first tells you similar proteins which have model structures, so which have experimentally determined structures, and similar proteins which do not have structural data in the PDB. So the first will have PDB codes associated with it. The second will not have any PDB information. This gives you, so again, we have information about the coverage. All of these icons have different information about the number of PDB structures where this related protein is present, if there are ligands in these related entries, and if there are any other proteins, interacting proteins present in these particular PDB entries. And the final tab is the publications tab. If you want to know more about the publications, we have listed them all for you over here. It's one comprehensive page where you can go to look at PDB publications, related reviews, publications, even UniProt publications. They're all present over here. Right. If you want to know more about the aggregated views, if you want to programmatically access them, we can definitely have discuss the possibility of future webinars targeting towards APIs. But there is a lot of information out there. We have webinars that are hosted. If you need to know more information, please let us know. We'll stick around for the questions. And yes, I think I'm doing well on time, right? Okay. So I think yeah, I've gone through the major part. I will hand over to Paulina for her bit on the AlphaFold database. And yes, thank you all so much. We will be very happy to know what you think about our resources and the feedback. Thank you. I'll stop sharing now.
Is everything all right? Yeah, so should be working now. Hopefully everyone is able to hear me over there. Just
Yes,
thumbs up if you can hear. Yeah, that's great. Thank you. Hi, I'm Paulina Magaña. Thanks for the introduction earlier. I'm a bioinformatician and I'm part of the AlphaFold database. Just to make things a little bit clearer, I guess you've heard of MLTB. So MLTB has many resources, and part of that is the PDB. But the PDB is one group, let's say it that way, that is part of EBI. And within that group, we have many resources. One of them is obviously PDBE, PDKB, but AlphaFold database is also part of this big resource. So it's a resource within a resource. That makes more sense. So moving on, I'm just gonna mention today about the AlphaFold database and introduce another resource, which is called 3D beacons. And let's start. So I guess you've heard about the structure prediction problem, which actually can assess that you should be able to predict the structure from the sequence. So we're trying to go from the sequence to structure. But before we dive into how this was solved in 2020, we will have to say first the Anfinsen's dogma, which is like the sequence should determine a structure, and we should be able to compute it this way. But then we have the Leviton's paradox, which is like, yeah, you should be able to have the structure from the sequence, but to do it that way, if you try to compute it, there are too many possible conformations, and they will take a search algorithm will take longer than the universe. This is why many bioinformaticians and computational biologists started trying to solve this problem. They developed something called CASP, which is the biannual critical assessment of structure prediction competition. They don't really like to call it competition, it's more of a challenge. And in 2020, AlphaFold came up, AlphaFold 2, but from now on, I'm just calling it AlphaFold, which was developed by Google DeepMind, and it can create highly accurate predicted models using just the amino acid sequence. So what is AlphaFold? I guess you've seen this diagram before. There's too many things to say about this. From the user's point of view, you only need the input sequence to be able to run AlphaFold. There are different AlphaFolds nowadays. You can use the source code, you can use ColorFold, and there are other ways that you can run it too. So the first thing you're gonna need is the sequence, and then from the sequence, what is happening in the under the hood is that AlphaFold is computing an MSA which contains sequences that are evolutionarily related to your sequence target. And why is this important? It's because for protein coding genes, when one residue mutates, another compensatory mutation follows. So you can see this from the MSA, and you can measure the coupling for any given residue pairs. So for those proteins that we don't have structure, the coevolution, the measurement of this could be used as a predictor of what residues are more likely to be in contact. AlphaFold can also use templates from the PDB, but it actually works well if the MSA is deep and has more than 30 sequences as part of the MSA. So then for the second part that they call Evoformer, this is the MSA is translated into a MSA representation, and the sequence, there's a pair representation for the sequences. But the Evoformer is doing something in computational science. Now, a couple of years ago, it came out the transformer, which the key source there is attention. So the objective of this is to identify which parts of the input are more important for the, in this case, for predicting the structure. In other words, it tries to identify the input that it should pay attention to. The difference between this AlphaFold is that there are two transformers actually. So one is for the MSA representation, and the other one is for the pair representation. And within these blocks, what is happening is that the MSA and the pair representation, let's say like they talk to each other in a way that they update the information. So at the very end, then there's the structure module which tries to summarize everything from the Evoformer and then have a 3D structure. Something that changed from AlphaFold 1 is that in this case, AlphaFold is learning from itself. So it doesn't just happen once, it happened 3 times. So it computes the one structure, and then it goes back and trying to update the information from the first recycle. The strength of AlphaFold, it's very good starting point for hypothesis, and it's been mainly used for X-ray and cryo-EM. It's only based on the amino acid sequences, and it's, if available, you can use templates, but it might discard them and use only the MSA if there's enough information coming from it. You also have the PAE, the PLDT, and the predicted atomic coordinates, which Sudakshina already mentioned, the PDB files and the MC files. So now let's talk about how can you interpret then these structures from AlphaFold. One of the things I like to say is that nothing in biology makes sense except in the light of evolution. But if we were to translate this into AlphaFold, then we would say nothing in AlphaFold predictions makes sense except in the light of confidence metrics. So whenever we have predicted this structure, there's going to have to be a way to interpret this. So the first one that we have is the PLDT. So this is an amino acid level confidence, or PLDT, which actually means predicted local distance difference test, or PLDT, and it comes from LDDT that has been around and has been used for assessing models. And roughly speaking, the LDDT is the predicted interatomic distances rather than how well the full predicted and the structures align. So this is telling you more about the local regions of the protein. And for AlphaFold, it goes from very low, which is orange, to the dark blue, which is very high. So whenever you're working with structures from AlphaFold, we usually tell users that go from try to use high and very high. Low values of PLDT have been related to the disordered regions for proteins or flexible regions, and that's something that needs to be also assessed when you're working with structures. Moving on now to PAE. So we were saying that PLDT is local, so PAE is a little bit more global, and this is actually a pairwise for the residues. So we have the residues that are aligned to itself. And the color green that you see here indicates the expected position error at a residue X if the predicted and true structures were aligned on residue Y. And what is measuring here is rather the confidence between 2 residues. So for example, we're trying to see how confident AlphaFold is that the residue 200 and the residue 600 are supposed to be close in space. So the greener it looks, the dark green it looks, the better. So here we can actually see that it's confident in this region and these regions. I'll explain a little bit of the diagonal, and it looks better when you see it in a GIS. So in this case, it's telling you more of like globular domains. So it is confident that these are supposed to be to defined globular domains, whereas when you go off the diagonal is the confidence between these 2 domains. So we see that in this case, it's kind of like whitish. So when it looks like this, it means like it's not really confident that these 2 domains in space are interacting or close together. It's important to use both the PLDT and the PAE. Don't try to use just one of them. Again, in this case, we can see that the PLDT is very low for that region, and you can start to speculate and say that by looking at it, it might look like that helix has a function. It's important to assess everything first. First of all, the PLDT is very low. Looking at the PAE, AlphaFold is not confident that that region is supposed to be located inside the donut-like. As many softwares, AlphaFold also has limitations. It can only accept the 20 standard amino acids. And by default, if you are running AlphaFold, you can only have 5 models per run. So if you were to try to predict different conformation, you will have to run AlphaFold several times. You can't do assemblies, but there's AlphaFold Multimem. It fails to predict the effect of mutations, but nowadays we have alpha-missense. It doesn't have the ligand molecules or the PTMs, or there's also another resource that is called AlphaFill. It's not designed for nucleic acids, it's just for proteins, and it won't model protein DNA or protein RNA complexes. Although a couple of months ago, AlphaFold 3 came out. I won't be talking about AlphaFold 3. Again, for that, I think we can take another time and talk about the newest release. One thing I want to highlight is that if you are working with transmembrane proteins, just be aware that, well, you need to know that AlphaFold is not aware of the membrane plane. So it's very important when you're working with these proteins to actually assess the predictions in this way. Also for antigen antibodies, it can predict them, but if it's gonna be good, that's the question. Because again, going back to the MSA, antibodies are very variable, thank you. So it's gonna be difficult to predict, to have a well-predicted structure. The same applies with orphan proteins. So where to start? You've seen AlphaFold everywhere by now. So we have kind of like a way to start, like a workflow. The first place will be the database. Then if the structure that you are looking for is not in the conformation for your research, you can go and use Google Colab. Or perhaps the protein that you're working with, you need it to be in complex with another protein. So for that too, you will use Google Colab. But it's a starting point to go to the database, assess the structure, and then go to the Google Colab. If you need to compute more, because the Google Colab relies on Google's GPUs and CPUs, although you can pay for a subscription, I think. I don't know how pricey that is, but I think it's quite pricey. The other option will be for you to run the source code. So what types of data do we have in the database? We have more than 200 million proteins. We have the PAGs, the PLDT, the PDB files, and the MC files. What we don't have yet are multiple conformations for the proteins, paraproteins, isoforms, assemblings, variant structures, and ligands. How can you search in the database? We have different ways to search. You can use the gene, you can use the protein name, you can use the UniProt accession or the organism if you rather see all of the proteins that we have that one. We also now have sequence-based search, which works with BLAST, and this is how it looks when you do the sequence-based search. How can you access? There are many different ways to access the data that we have in the database. The first way will be just go to the website, use the API if you are a bioinformatician. That'll be the best way to retrieve more than one structure. We also have the FTP. For the FTP, we have them as proteome, so you need to download the whole proteome. Are really useful if you have to use the whole organisms, for the whole proteins for the organism. There's an extra way that you can access to, and it's the BigQuery. For this, you're gonna have to use a Google account and just connect to the bucket from GCP. The free tier allows you to query like 1 terabyte, but this is a good way to start. If you need to download several proteins, but you don't want to use the API, you rather use the BigQuery, and it doesn't take too long to run it, to be honest. The database has been growing since its inception in 2021, and now we have improved the results that you get for filtering, the PAE, the 3D visualizations, and last year we added the sequence similarity-based search and structure similarity clusters. I'm gonna talk about this just now on the next slide. So the AlphaFold has more than 200 million proteins. And for this, we collaborated with Martin Steniger. He has his lab is based in Korea, South Korea. What his lab did is they cluster the whole AFDB, the whole database, firstly based on sequence, and then they used the representative for all these clusters and cluster them then based on a structure using FOLSEC. And that's how we ended up having 18.8 million clusters. And we made them all available in the database in this way. So we have them in the structure page. This is how they look. You have the 3D viewer, the PAE, the PLDT coloring. And you can see that here we have a table for all the structures, in this case, that were clustered based on sequence. And if you go to the next tab, you see the ones that were clustered based on structure with FOLSEC. We have another resource we added recently. I think it was May. I believe it was in May. It's AlphaMesens. So AlphaMesens is out now. And how AlphaMesens works. So they use a bit of, it was also developed by Google DeepMind, and they use some parts of AlphaFold, a protein language model, and they use some ways trying to predict if one variant is benign or pathogenic. The way it looks is like it produces a score that goes between 0 and 1, rating the likelihood of the variant. To classify it, for this bit, what they use population, and it's a weekly supervising on human and primate sequence variation. That's how it works. So it's aware of the context in the protein structure from AlphaFold, and it's also aware of the variation of residues in MSAs and sequences. Just have to highlight that this is only for humans, and you don't get a structure of the variant. You rather get a score of how likely that specific variant is pathogenic or benign. It makes more sense when you see it in the database. So we got this data and we put it in the database for the users to make use of it for their research. And we have it in two different ways. So the first one will be the 3D viewer. Within the 3D viewer, you can toggle between the PLDT and the AlphaMesens score. For this one, for the 3D viewer, what you're looking at for the colors is actually the average pathogenicity. Or if you were to use the heat map, in here you can actually see what will happen if you mutate one specific residue for the one specific residue to another, like valine. You get one score and you get the variance, and we also have the coloring based on pathogenicity or benign. If you decide, you know what, this filtering, I want to just focus on the ones that are very likely to be pathogenic, you can just go on and change this filters. So we made this available for users to be able to change it according to the research. If you prefer working with the data that's coming from the genomic builds, you can download it from the HG19 and the HG38. Or you can also download the data that we use to have this heat map, so you can recreate a heat map for your research. So I've been talking about structures, how good they are, how we have all this data, the amount of data we have. one of the questions that we always get is like, can I use them? Are they available? They're actually really good for hypothesis. So this is a paper that came out this year, but it was used to be a preprint, so this is finally out. So this is one study by Tom. Won't try to pronounce his surname. I'm so sorry. And they took the newly determined experimental structures from the PDB and then compare them with AlphaFold. So they found that for the main change, the RMC was about 1 angstrom versus 0.6 for experimental structures, which is very close if you're using the structures from AlphaFold. They also found in terms of the PLDT that when you have a PLDT that's more than 90, then the RMC is 0.6 angstroms, which is very close to the experimental resolve structure. As we mentioned before, as I mentioned before, for the PLDT that's lower than 70, then the median RMC goes higher. In terms of the side chains, they found that 20% of the side chains are substantially different, but that also means that 80% of them showed a perfect fit. Don't get too. I know sometimes we like to see statistics and like, oh, it's 20% error. Yes, but it's 80% good fit. In the same way, for that 7%, 93% of the side chains are roughly correct. You can also use this if they do. You can also use the lab and have to be just for trying to solve structures. So in this case, for this paper, they wanted to understand how mutations will impact the catalytic activity of this protein. They mutated them to alanine, and they found that it abolished the protein expression. And they mutated them to serine to test the hypothesis that an intermolecular serine hydrogen bond might be able to stabilize this protein, and they actually failed to restore expression of it. Moving on now to 3D Beacons. 3D Beacons is an open collaboration, is another resource, and as Chatterjee was mentioning earlier, we bring in information from different providers. So in this case, we bring structures from different providers, and it's trying to provide the coordinates and meta information from all the different data providers, resources in more standardized way in a platform. Everything is free for this resource. Is CC BY 4, and it's an open collaboration. You can find structures from UniProt accession, sequence search, and if I remember correctly, by ensemble transcript too. This is how it looks when you go to one of the pages from 3D Beacons after searching. So we do classify the structures based on if they are formational ensemble, templates, ab initio, and experimentally determined structures. The best way to access 3D Beacons is actually through the API. So as bioinformaticians, we like to access everything through APIs. So we also have them for 3D Beacons. For the sake of time today, if you want to learn more about AlphaFold, we recently developed a practical guide. It's an open training that we developed with Google DeepMind. It's freely available, and that's the QR code where I dive more into how you can access the structures, access them, identify the best way to access the database, if you need to use the source code, colorful, and many other things that we cover in the training. If you want to get more updates or what the PDBE is doing, what we are going to release soon, we have the X, aka Twitter, and LinkedIn too. This is part of our team. It's not the whole team. There are some missing with the photos missing here. And our group leader, Samir, Blanca. And lastly, we welcome the feedback for this webinar. As Sudakshina mentioned too, we understand that there was not enough time to cover all the resources we have, everything you can do with PDBE, PDBE-KB, how you can access 3D beacons, AlphaFold DB, or AlphaFold itself, and how you can use it in your research. So we're thinking that perhaps we can have another webinar or workshop in the future. So if you like to provide your feedback, that's the QR code for that.
ये QR कोड अलग है ये QR कोड अलग है
I'll just gonna leave that. Thanks.
And thank you very much, Dr. Paljana. And it's time for questions. Aditi, please.
For specific type of analysis such as cryo-EM data visualization. Yeah.
Massive and built to view really large structures and all the information, all the map files together. So you should be able to view, and you don't need an extra plugin. Please try it out.
Yes, I just wanted to add that too. So Molstar also works as a website itself, which is more as a web app. So you can actually bring data from PDBE and from AlphaFold. I just shared the link for that. And
so that's basically the Molstar page that you can access outside of PDBE. So suppose if you have a PDBE or MNC file that you have generated and you want to visualize it, you can use this particular link that Polly has shared to upload the file and visualize it using Molstar.
Yeah, thank you. I hope this is
So next question we have is Abdul Munafi is asking, he's thanking you for the wonderful presentation first, and then he's asking that from your last statement, you said what procedure do I follow for understanding the AlphaFold? I think basically he's asking the training which you showed for AlphaFold, the link to the training for that. So I think it's yeah.
it's been used, how you can use it, and other things that are going on at the moment. So just stay tuned because we are gonna update it soon.
thank you. From the questions we received,
Registration form. One of the question is that many of the times it gets difficult to find active site residues of the protein because of its unavailability of PDB structure. So is there something we can offer to resolve this?
I really understand the question. I mean, do you have a computed structure from AlphaFold that you're looking at?
I think
the question just mentions that PDB structure is not available. That means it is not submitted in PDB. So that means it is a predicted structure, right? So precise structure is not available. So I think the sender is asking some help on that. So maybe we can get back to this question if we have more information.
Going. मैंने सुना ही नहीं, मैंने ऑफ कर दिया ना, इसलिए मैंने ऑफ कर दिया, इसलिए पता नहीं चला मुझे।
The database, the second step will be to use colorful. You might need to play around with the MSA and the seeds for these predictions, because what you want to predict is conformational variability rather than the same conformation over. And then you might have to use P2Rank to predict one binding site for that, for those structures.
Another question we have is that is it possible to do docking studies with AlphaFold?
It's a very similar answer to what I just said. It will depend again on the conformation of the protein that you have. There are a couple of papers that have successfully docked molecules for screening. I can get the papers and send them over to you in an email.
Thank you. Do we have time? You have. Yeah.
Samrat asking a theoretical question that is the full structure of the protein predictable by only amino acid sequence, or do we need to also have the direction of the translation? He must have read that the rate of protein folding is much faster than the rate of translation. So, will the structures of two amino acid sequences be same if they are translated in opposite directions? For imagination.
Direction, if it's 5 prime, 3 prime, minus and yeah. But you only need the sequence for the variability, and you can vary that with MSA and template. So I mentioned one of the things that you can play around is also MSA and templates. So if you block AlphaFold, or rather colorful, to not compute a big MSA and compute a smaller one, and then you template from PDB, you can try to force AlphaFold into predicted one structure that's similar to that template. So it will work more of a like template modeling.
Yeah, thank you. Another question we have is from Dr. Sohi. She was also one of our workshop instructors in last year. She's from the University of India. So she's thanking both of you for the wonderful session. And then the question is, are there any PDB quality score or slide card type metrics to assess and choose the best cryo-EM structures for a given protein, especially in the context as cryo-EM structures have varying local resolution, which can quite be different from single global resolution.
Do not use the term best cryo-EM structure, because we are a database and we cannot determine what's best. We can only show you what the scores are, and ultimately you are the one who has to determine if it's good enough, if it's better than the others. And we are aware that in case of cryo-EM structures, there may be local versus global resolution. We are working with the EMDB, which is the electron microscopy data bank developers, and who are experts in cryo-EM as well. We are trying to develop more metrics and quality controls to at least showcase these differences. That is on our agenda, but yeah, we'll keep working on it. I don't think there's anything right now that will help you differentiate between local versus global resolution.
Thank you. Yeah, I think that
questions. The only question we have left from the questions which came in registration form is that is there any way to find a PDB ID of novel proteins? So I think it is in
To the PDBE search page. Probably do you want to share your screen for a second? Yeah. So if you go, so this is our search page. Do you see those tabs called latest biology, latest chemistry, and latest entries? Those are all listing novel proteins. So if you click on it, there are only one instances of these proteins. They are new to our database. So how do you know they're new? If you click on the PDBE KB tab, so you see there's only one structure where that's what it's showing. Now you have to remember that these structures change because we release new structures every week, so we will keep updating them every week. So yeah, this is a local, a really quick way to look for novel proteins every week. But if you are really interested in a particular protein, KB and search for it, the PDB KB page will really tell you, at one instance, how many structures are present in the database. And same goes for the tab for looking for novel ligands as well. You will get all that information.
and rest of the questions deal with how to use PDBE for a research, and I think both of you went through it very well in the presentation. So with that, and Roshni in our chat box is asking that please cover more sessions like this and have softwares and other use cases. So yes, Roshni, please fill out the feedback form and there's an option for it so that we can request Dr. Ganguly and Dr. Mutana for a follow-up workshop. So yeah, with that, the question session ends. I request Govinda to please take over.
Thank you very much, Dr. Mogana and Ganguly, for this wonderful session. I think the resources we have gone through, the PDBE and AlphaFold, these are the two resources which have been used by the researchers in daily life. The thing is, we most of them don't know about the resources, when to use, and what is the use in the real time. So I think it is a wonderful opportunity for everyone who is looking for some of the web servers or some of the databases that we can access very easily. So these tools are these databases and the resources which we have seen today are easily accessible to everyone, and there is no programming language, any type of technicality is, there is no obstruction for everyone. So even the person with the no programmatic experience will use these resources very easily. And thank you for giving these lectures. And we have wonderful volunteers in RSG-India, and they have made one they have one message for you from RSG-India. So ISCB to Dr. Magana. So ISCB RSG-India is delighted to acknowledge your significant contributions to the field of bioinformatics. With Protein Branda Bank, your dedication and perseverance in scaling these areas niches are commendable. So your commitment and expertise are truly inspiring, and we are honored to have you and participate in this event. And we believe your valuable insights and guidance will ignite the passion for proteins and their structures among young researchers. And thank you, Dr. Mogana, and the same goes to Dr. Ganguly. And thank you both of you for having this session with RSG-India. We hope we hope RSG-India will be in collaboration with you for the more workshops in upcoming future. And we have already shared the feedback form with the volunteers, and if anybody left to fill the feedback form, please scan the QR code here, and also I shared the link in the chat box. So please wait at least 15 days to receive your certificates. And Thank you very much for everyone. So at the last, I would like to ask everyone to open your camera so that we can have a group photo. Yeah. Please turn on your camera so that we can have a photo. Yeah. So I think from next time we should have done this earlier. Like at the end of the session, everyone will do this. Yeah. Thank you, everyone. I think, yeah. Yeah, the photo is done. You can leave now. Yeah.
To be here. Many thanks for having us. Again, we welcome the feedback for the workshops, and we're already looking forward for the next one.
Yeah. Dr. Ganguly, your mic is off. Yeah.
Oh, I thought it was. Can you hear me now?
Yeah.
No, yeah, it's been great, and thank you for the opportunity to come and talk. Hopefully, we'll get in touch for more sessions in the future.
Yeah, sure. So we're going to share this feedback form with the all the participants in the email so that we can receive the responses. Yeah.
Thank you. That'll be great. Well, everyone, thanks for coming and have a nice weekend. It's Friday.
Thank you, ma'am.
Thank you.
Bye.
I would like to thank Dr. Sohini for joining. Ma'am, are you available for 2 minutes for us to talk? छोड़ दिया क्या? सोहिनी मैम. हाँ, उसमें RSG इंडिया लिखा हुआ है. फीडबैक फॉर्म देखा है? मैंने देखा था, फिर हमारा और उनका, फिर उन्होंने शेयर किया, हमारा शेयर किया, फिर अलग अलग होगा इसलिए मैंने उनका शेयर कर दिया. उनको पूछ लेंगे कि ये देखो ICB-RC इंडिया लिखा हुआ है, तो उनसे मांग लेंगे ना, एक्सेस मांग लेंगे. Okay, thanks everyone for joining, and I think please fill the feedback form. We may have the based on your feedback, we may have the workshop with the same team in future. And Ajay, I don't know, like, okay, are we expecting have to the file for the presentation? So we will request speakers. If they provide, then we will share it with everyone. Okay, everyone. Thank you for joining. Bye bye. ठीक है. अच्छा.
अल्फा फोल्ड वाला मेरे साथ से वही वाला लिंक है जो RCB वाले ने शेयर किया था. जो RCB नहीं कहते आप लोग. वहाँ जो वीडियो वाले ने शेयर किया वही आता होगा।
हाँ वही है, उसी टीम से है ना ये अटेंडेंस का तो आ गया, 83 लोगों ने किया था तो ये तो चाहिए हमको है ना।
ऐसे आ गया ना अटेंडेंस टैग, ठीक है फिर वैसे
तो फीडबैक मांगूंगा अभी
50% इसका ज़्यादा होगा उसको भेज देंगे।
50% आई थिंक लोग लेट भी ज्वाइन किया, जिसका भी 20% से कम है ना उसका हटा देंगे।
20%, ठीक है।
20% से कम है
तो इसकी अभी एक्सेल शीट बना दो ना।
वही तो मैं बोल रहा हूँ, फीडबैक का ज़रूरत ही नहीं है, किसका 20% से ज़्यादा है उनको दे देंगे।
और मांग लेना दीदी को,
नहीं नहीं मांग ले, वो तो देखना भी है हमको फीडबैक फॉर्म।
ये मुझे हम तीनों को मेल कर दो अभी कर
PDF
दो।
उसमें शेयर कर देता हूँ ना,
इसमें
ड्राइव में अपलोड कर देता हूँ।
अच्छा वो RSG वाली।
अरे यार ये प्रिंट हो रहा है क्या? हट यार। PDF
कितना बुरा है, एक तो इसके फैंस इतने ज्यादा है। हेलो गोविंदा,
हाँ वही कि वो
तूने नाम नहीं दिया।
जिस बंदे ने लास्ट तक कैमरा
अब्दुल
ऑन
जो
किया
था,
था मैं बोल भी रहा
वो भाई पीछे दूसरा भाई आ गया ऐसे ऐसे करके गया है, देखा था?
वो मजा
ये भी नहीं देखा, ये भी नहीं देखा, ये भी नहीं देखा इसने यार, तूने भी नहीं देखा
क्या?
ये
ये नहीं देखा
ये भी नहीं देखा।
ये किसने
उसके पीछे भाई आगे कर रहा
अच्छा,
है ये।
अच्छा वो बंदा तो कह भी रहा था हेलो गोविंद,
हाँ
ये पीछे
वही
आ गया।
तो।
सच में बोला वही को जानता है।
मैंने ध्यान नहीं दिया कौन है वो।
नहीं
तू
बोला
entertain
मैंने
नहीं कर रहा।
बताया भी, मैंने फिर पहले सुना नहीं आपने।
मुझे लगा तूने इग्नोर मार दिया उसको,
मुझे भी
मुझे भी लगा। होता है ऐसे होता है,
वो इंडियन था कोई।
बड़े लोग ऐसे इग्नोर मारते हैं।
एक हो गया तुम लोग का,
हैं?
हो गया।
नहीं अभी कहाँ। वो भाई इतनी शिद्दत से बैठा हुआ था, इतनी देर से वो ऑन करके कैमरा ऑन करके तेरे लिए।
हाँ बहुत देर तक उसने क्या है, जब
हाँ।
तक लीव नहीं किया तब तक कैमरा ऑन था उसका।
हाँ।
अरे वो यहाँ पे था नहीं क्या, यहाँ पे साउथ अफ्रीकन बंदा, वो नाइजीरियन।
अरे वो स्क्रीनशॉट लिया होगा ना तूने अभी?
हाँ।
लिया है ना?
स्क्रीनशॉट करो।
हाँ।
उसने लिया,
मैंने भी
हाँ
लिया।
मैंने भी लिया शायद।
मैंने भी लिया।
एक मिनट कौन है?
ये ये तरह
ये वाला
अच्छा।
ये वाला भाई कह रहा था हेलो
उसके
गोविंद।
पीछे वाले नहीं, पीछे वाले ने
अरे
बोला
ये
था।
पीछे वाले ने
प्रशांत
ये
है
किया
यार।
था, इसने तो वो किया था।
अरे ये प्रशांत है, वो शायद यूनिवर्सिटी का है।
हेलो गोविंद।
साउथ यूनिवर्सिटी में से
वो नाइजीरिया से है।
गोविंद से कितना भाव बढ़ेगा।
कोई नहीं बोला, ठीक है, इससे कुछ नहीं होने वाला।
Do we have room for TIL? नहीं, not TIL. वो वाला
अरे
था।
अब जो
TechX.
करना है करवा लो।
Do we have room for TechX?
कर दो अगले
है?
महीने।
अगले महीने तो नहीं, मतलब एक सर है मेरा, उससे पूछना होगा।
अभी जुलाई का हो गया ना।
He is in NIT जबलपुर, assistant professor. पूछ लेती हूँ एक बार। कल ही पता चला मुझे कि उन्होंने एज अ फैकल्टी ज्वाइन किया यहाँ पे। बोला 1 साल हो गया मुझे ज्वाइन किए, मैं आपको बताया बिल्कुल भी।
अब हमको लगता है कि TAL करना चाहिए।
टीचिंग इज लर्निंग जो हम अपने वाले करते थे।
TL एक शूट करता है और टेक्स भी करता है, दोनों का।
ठीक है।
लाके उनको मेल भेज देता हूँ, फीडबैक भेजना है जो करना है बस उसके बाद।
टीएल हमें अपने से
तो फीडबैक हम लोग खुद बना दिया।
किस नाम से
ड्राइव में उसमें MLTB का फोल्डर है ना, उसमें PDB का फोल्डर है, उसमें अटेंडेंस का फोल्डर बना के डाल दिया।
CSV फाइल है,
CSV फाइल PDF दोनों डाला।
फिर CSV वाला प्रिंटर मार 20% से आता है।
हाँ वो यार आती थी इसमें, लोग ना 20% से जितना कम टाइम में ज्वाइन किए उनको हटा देके सर्टिफिकेट नहीं देते।
20% भी करना है।
तो देखो हमारे सेशन एग्जैक्टली 1.5 आवर चला था, उसमें 20% मतलब कितना है?
कितना में चला?
1.5 आवर 90 मिनट्स
90 मिनट्स का 20%
मान लो, सो
80 मिनट
कम
80
मिनट कम
80 मिनट
यानी 1 घंटा और 10 मिनट बैठा हो तो उसको अटेंडेंस मिलनी चाहिए।
ठीक है 20% रखना है, 20% और 20% ज़्यादा
20% से जितना कि हाँ 20% मिनिमम कर दो। उनको बोल देंगे अगर कोई सर्टिफिकेट मांगा तो
मांगेगा तो दे देंगे,
नहीं मांगे तो बोल देंगे ना, अगली बार पूरा करो एक। और
तुम दोनों बड़ी शिद्दत है, थोड़ी देर में बैठे रहते थे मेरे पीछे, वो काम कर रहे थे, मैं बोला कि छोड़ो, फिर उनको जनता प्रकट हो गई और उसी बीच में उनको वेंडर को भी आना होता है, सबको जनता प्रकट होना होता है। अभी भी जब इसके लिए बैठे थे ना, लास्ट में कैमरा ऑन कर एक बंदी आई कहती मेरा ना ये वो डिस्चार्ज हो गया, इसको चार्ज में लगा के छोड़ दो, हेडफोन को चार्ज में लगा के बैठी है, हेडफोन कोई वेंडर है, बहुत बढ़िया। तो चले गई थी ना ऑल आउट, अभी उसको ऊपर तो वो भी टूट गया अभी, फिर उसमें छेद करके सिरिंज से उसका सारा माल निकाल के दूसरे वाले में डाला है, तब जाके उसको लगाया। फिर ये सारे कांड अच्छे में लगते हैं। आकृति, कल नीले को मैंने बोल दिया, नीले ने पहले मुझे बोला कि हम ना एक काम करते हैं, जितने भी स्पॉन्सर्स वाली कंपनी है जिनको हम टारगेट कर रहे हैं, उनका हम ना जो कंपनी का प्रोफाइल लिंक्डइन है इन सब पे उनको टैग करना शुरू कर देंगे। मैं दिस इज अनएथिकल, कि आप बिना परमिशन के बिना कंसेंट के आप ऐसे करोगे, दिस इज अनएथिकल। तो मैंने बोला कि अगर कोई बड़ी कंपनी है, अगर उसमें केस वगैरह करते हैं नोट विल डू ना, ऐसे आप किसी को वो थोड़ी करोगे, लाइक नहीं वी वर डिस्कसिंग आउटरीच एंड ऑल, तो वी थॉट कि ऐसा कर सकते हैं। हाँ, बिना पूछे कैसे टैग करेंगे। फिर दूसरा उसने जब वो वाला बोला कि हम ओकिनावा की म्यूनसिपैलिटी या फिर बाकी वो जो ऑर्गेनाइजेशंस हैं उनको कांटेक्ट करते हैं, और उसने मुझे बोला कि निकिता प्लीज ट्राई फॉर एटलीस्ट आवर ओटीज, फॉर ऑल द ओटीज आई शुड ट्राई। ठीक है, नहीं तो फिर मैंने उसको बोला कि व्हाई ओकिनावा विल स्पॉन्सर अस, फर्स्ट ऑफ ऑल वी आर नॉट फ्रॉम देयर कंट्री। ठीक है, क्योंकि देयर इज अ रूल दैट एवरी गवर्नमेंट हैज टू फॉलो, एंड अकॉर्डिंग टू दैट उनका पहले से स्पेसिफाइड होता है कि उनको किस इसके लिए कितना पैसा यूज़ करना है, एंड दिस इज़ अ वेरी शॉर्ट टाइम। क्योंकि इंडिया में भी अगर कोई एक्सचेंज प्रोग्राम होता है या फिर कोई ऐसी फेलोशिप होती है, दे मतलब टेल पहले से ना कि ऐसा कुछ है, यू अप्लाई फॉर। ठीक है, ऑल ऑफ अ सडन यू कैन नॉट टॉक टू देम। ठीक है, और दूसरी बात, इट इज़ बेटर कि हर ओटी अपनी कंट्री के उससे फेलोशिप या फंडिंग बॉडी से बात करे, so that we get to know कि अरे तुम्हें मिल रही है कि नहीं मिल रही है।
कमेटी में तो फाइनेंस कमेटी में है, तेरा काम सिंपोजियम के लिए स्पॉन्सरशिप कलेक्ट करना है, ना कि ओटी।
नहीं, she was
that is one part of it, that is not the only।
नहीं तो उसने जैसे मैंने बोला, मैंने उसी समय उसको बोल दिया कि मतलब दिस इज एवरी ओटी शुड डिसाइड ऑन देयर ओन।
और कौन से ओटी जो मैसेज का रिप्लाई नहीं करते वो वाले?
तो उसने फिर उसको बोल दिया कि भैया इसको हटाई दो तुम, वो लिख रही थी उसमें एजेंडा में, मैं हटा दे इसको, मैं दिस इज नॉट माय रिस्पांसिबिलिटी। ऑब्वियसली मैं तो यही सीधा क्वेश्चन पूछा कि व्हाई अ गवर्नमेंट विल स्पॉन्सर अ स्टूडेंट फ्रॉम डिफरेंट कंट्री, व्हाट इज देयर बेनिफिट?
क्यों तो महान है कि बीबीसी के लिए स्पॉन्सरशिप मांगती है, तो
फिर वो पता नहीं प्रदीप से किससे बात हुई थी, मतलब प्रदीप ने सजेशन दिया कि हम अपने मर्चेंडाइज बनवा के हम उसे बेच लेते हैं। प्रदीप ने सजेशन दिया ना, देन शी वाज आस्किंग मी कि उसे कैसे बेच सकते हैं, कहाँ बेचेंगे? जो मतलब जो इसको अटेंड नहीं करेगा ASCS को, उसी को तो बेचना है बेसिकली, तो कहाँ बेचेंगे? मुझसे पूछ रही। मैं APBJC हमारा मेन इवेंट है ना, वहीं बेच लो, बोलो कि एक कैनोपी वहीं लगा लो और बोलो जिसको खरीदना है APBJC के वो मेंबर वहाँ पे आके खरीद लो। मैं एक तो पहली बार क्यों खरीदेगा कोई?
ये केसेस पहले इसके बाद में
हाँ तो वही मैं कह रही हूँ ना,
चलोगे क्या?
भगवान जाने।
वो
मैं एक काम
वाले
करूँ?
दिन बेचने को तैयार है।
फिर उसने पूछा कि मैंडेटरी और नॉन मैंडेटरी कौन सा है। मैंने बोला सब कौन नॉन मैंडेटरी, इसमें मैंने लिखा है आपकी मर्जी है, दो चाहे मत दो, है कि नहीं? इट्स योर चॉइस टू गिव और नॉट, नोबडी इज गोइंग टू आस्क यू। क्योंकि जो उस दिन प्रदीप ने बोला कि they are sharing their knowledge, they will get to know a lot of things, so why they need any memento or any of this. If you think like this, then there is no point of having any money. Why are you are worrying about money ना? then क्या करोगे उस पैसे का? क्या डालोगे? खाने का आपका सारा हो गया, वहाँ का आने का आपका सारा हो गया, फिर क्यों चाहिए पैसा? किसी को जो वो आएगा, गेस्ट आएगा या जो भी पैनल आएगा, स्पीकर आएगा, उसको आप कुछ देना नहीं चाहते, फिर क्यों चाहिए पैसा तुम्हें? ऑब्वियसली, या तो आप ऐसे ब्लंट स्टेटमेंट मत मारो। उससे सारा मैंने उसको बोला ये मैंडेटरी नहीं है, ये मैंडेटरी नहीं है, ये मैंडेटरी नहीं है, हमने सब नॉन मैंडेटरी में डाल दिया। वो एक पेन और एक नोट पैड, उसको मैंने छोड़ा, पहले इसको तो मैंडेटरी में डाल दी कि कम से कम इतना तो कर लिए। तो बस वही चीज़ उसने मैंडेटरी में डाल के छोड़ दी। मैं होता तो किसी के जेब से भी निकाल लेंगे। 120 बंदे लिखे तो उसमें 100 कर दिए, ठीक है 100 कर दिए, तो एक काम करो 100 के साथ
चलना शायद 100 लोगों की।
हाँ जगह 100 लोगों की है। अरविंदा
बस क्या
फिर नीले सजेशन दे रही है। हमारे यहाँ पे तो ऐसा होता है कि हम किसी स्पीकर इसको पैसे दे देते हैं, हम तो किसी बॉडी को मतलब उसको देते हैं, एनजीओ वगैरह को पैसे। अच्छा एनजीओ को पैसे दे दो।
ये एनजीओ कहाँ से आ गए इसमें?
हम ही एनजीओ।
वो तो वहाँ पे बॉक्स लगा देना ना, चंदा कलेक्ट कर लेना, जो जाएगा जैसे सब जगह होते हैं,
कैंसर सर्वाइवर, प्लांट्स के लिए, हाँ पेटा हो गया, फलाना ढिंगाना, सब एक डब्बा डाल देंगे।
एक डॉलर, एक जापानीज येन सब लोग डोनेट कराना।
अपने हिसाब से चंदा इकट्ठा करो, हम बैंक में जाके पैसे डाल देंगे कि लो एक्सचेंज कर लो इसको पैसे।
अपनी करेंसी डाला, उसमें वर्ल्ड बैंक में ले जाओ फिर उसको डब्बे को।
चलो हमारे हिसाब से हमारा चंदा यही है।
कब स्टार्टिंग में आया? अरे यार मुझे पता नहीं था।
मुझे लगा कि अब सर रुकेंगे 2 मिनट तो उसके बाद बोल देंगे, अभी वो बोल ही रही थी, बीच में थोड़ी बोलेंगे कि भाई। और
अच्छा।
मुझे ये भी नहीं पता कि जब निकिता ने ये इसके इनके बारे में मतलब ये कोर टीम के बारे में बोला है तब वो सर थे या नहीं थे।
अच्छा।
पता ही नहीं चला कि कब चलेगा, ये नहीं पता। मतलब आएंगे मुझे 110% पता है,
वो तो अटेंडेंस में दी जाएगा।
अरे आएंगे, मैंने खुद देखा ना उनको, पहले टेढ़े होके बैठे थे पूरा टाइम, कैमरा टेढ़ा रख के बैठे ऐसे।
अरे वो बैठे हुए ऐसे ये दिखता है, उनका जॉ लाइन दिखती है जो कि है नहीं, वो दिखते हैं उनकी ऐसे
टेढ़े से सो रहे हैं पूरा अपना, उसके आगे बैठते बिल्कुल, क्या बोलते हैं वॉलपेपर उनके घर,
सौंदर्य
तैयार होके बैठते थे।
आया था
20 मिनट बैठ के गए
यार। ये क्या इतना जल्दी आ गया? नहीं 44 सेकेंड्स है यार, ये रहा। 1 मिनट के लिए भी नहीं, 3:10 पे आया था। ये क्या है?
वो ना आई थिंक वो ना ढंग से रिकॉर्ड नहीं हुआ, कब आए कब गए, 3 टाइमिंग पे दिखा रही है।
नहीं ये ज्वाइनिंग टाइम दिखा है इसमें, देखो ज्वाइनिंग टाइम इतना मैं
नहीं
ज्वाइन
नहीं
किया
नहीं,
था।
ज्वाइन इन्होंने तब किया और ये यहाँ पे चले गए, यानी 16 मिनट बैठे हैं क्या ये?
नहीं ये रहा ना 0 मिनट 44 सेकेंड्स ज्वाइनिंग टाइम दिखा है इसमें। मतलब देखो हमारा यही से शुरू हुआ, यही
अच्छा
खत्म
अच्छा,
हुआ,
हाँ हाँ
और इन्होंने यहाँ ज्वाइन किया और
हाँ।
44 सेकेंड छोड़
44
दिया।
सेकंड्स के लिए थे वो, कोई नहीं 44 सेकंड्स
तेरा देखता है, तेरा में पास हो जाएगा। ये रहा देखो 251 पे ज्वाइन किया।
अरे
देर
गोविंदा,
130
तूने जो 20 मिनट वाला बोल दिया है ना उसको कि 20% 20 मिनट के लिए जो था उसको नहीं देना, उसको ज़्यादा टाइम बढ़ा दे, क्योंकि हमने सेशन ही एक्स्ट्रा टाइम के लिए किया है। 16 मिनट तुम्हारे यही एक्स्ट्रा है, तो एक तरह से पूरा हो गया वो। जतिन को बोल दिया कि परसेंटेज ज़्यादा काउंट करेगा।
देखो अगर ठीक है करने दो, ये तो सही कर रहे हैं हम।
तो फुल सेशन की दे रहा है तू अटेंडेंस, इसका मतलब क्योंकि
देखो
20 मिनट
सेशन शुरू हुआ 4:15 से, है ना? सॉरी 4:15 नहीं।
गोविंदा यहाँ पे देख, सेशन जो शुरू हुआ ना, अकॉर्डिंग टू दिस सेशन यहाँ से शुरू हुआ और यहाँ खत्म हुआ, ठीक है? तो अप्रॉक्सीमेटली तेरा 1 आवर 45 मिनट्स का सेशन हुआ, ठीक है? उसमें 20% अटेंडेंस कितना हो गया तेरा?
50 मिनट्स ऐसा कुछ है। बट इधर देखो एक बार, सब लोग 3 बजे 15 ही ज़्यादा दिख रहा है ना इसमें। एक मिनट, यहाँ पे कोई शॉर्ट करने का तरीका है? इसको मैं खोलता हूँ एक्सेल में। यहाँ पे जो देखता है ना कि इसमें कहाँ है Excel? ये देखो, सारे लोग 3 बजे ज्वाइन किए मैक्सिमम, क्योंकि इतना लोग 2 बजे, मतलब 2 बजे से पहले किया था। बट ये लोग ज़्यादा ही किया था। देखो जो लोग पहले किया था वो लोग,
ठीक है,
वही 3 बजे के बाद जो भी जो आया था मोस्टली 3:10 के बाद ही आया था। देखो
ठीक है।
ये सब लोग 3:10 के बाद आया था। बट
नहीं, अब
जो
देखना, ऊपर जाना, ऊपर जा, ऊपर जा। जैसे यहाँ
ये
पे है
देखो
310 पे, कोई 20 मिनट रुका है, कोई 34 मिनट रुका है, कोई 37 मिनट रुका है, कोई 36 मिनट रुका है। इनको देखो कि दोगे कि नहीं दोगे, ये कह रही हूँ मैं।
ये 6 मिनट रुके, इसको तो ऑब्वियसली नहीं
ठीक
देंगे।
है, अब जैसे इसने ज्वाइन ही 3:30 बजे के आसपास किया है, तो मान के चल, ठीक है? तो ये 36 मिनट रुकी है।
हाँ वो वही मैं बोल रहा हूँ, 20 मिनट से ज़्यादा जो भी लोग हैं उनको दे देते हैं।
ठीक है,
ये जो अटेंडेंस है
हाँ हाँ,
20
ठीक है।
मिनट से ज़्यादा है उनको दे देते हैं।
फॉलो वर्कशॉप का भेजा है।
करवा लेते हैं।
वर्कशॉप नहीं है, वो है कोलैबोरेशन, अगर किसी को करनी है या किसी को आगे ट्रेनिंग करवानी है।
अच्छा, वो जो उन्होंने बोला था कि
हाँ, वो मैंने उनका आ गया, मुझे पता उनका रिप्लाई नहीं आया। तो ये फॉर्म में से मैंने डाल दिया। फॉर्म हम एक काम करते हैं, सबको भेज देते हैं बस, जो मतलब फर्स्ट एपिसोड के पार्टिसिपेंट्स थे, जिसको इंटरेस्ट होगा वो कर देगा,
मेल भेज देते ना।
ठीक है? मतलब यही था, यही डिसाइड किया था हमने कि हम मेल भेजेंगे, रिस्पॉन्स आएगा। उन्होंने 31 अगस्त तक ओपन रखे हैं। ठीक है, कर लेंगे। हमने तो फॉर्म बना।
ये बोल रहे हैं विशाल, इट इज़ पॉसिबल टू कन्वर्ट दिस फ्लायर इनटू मीटर।
RSG-India related question डालना है इसमें?
से बात करके आता हूँ।
आपने जो पीडीबी फाइल थी उसमें म्यूटेटेड ऑलरेडी थी ना, मतलब फाइल तो वो सेम पीडीबी फाइल है ना जो उसको मिस्टेक से रन करने की मिस्टेक में वाला जाएगा ना एनालिसिस नहीं तो फिर इवन आप अगर उसमें देखिए तो इनडायरेक्ट कोरिलेशन हर सर्वर का जो होता है मतलब जीरो एरर और पैरामीटर डेवलपमेंट होते हैं क्योंकि हमें नहीं मतलब हम इसको फाइनल कैसे दे सकते हैं विजुअल सीमेंट इट्स नॉट अ एस्टैब्लिश्ड मेथड हमारे पास जितने अवेलेबल हैं हम सबसे करके देख के फिर ये बताएंगे भाई हमारा रिजल्ट्स ये हैं तो अभी भी ये जो आए हैं वी कैन नॉट से विद 100% कॉन्फिडेंस ना कि ये है मतलब कोरिलेटेड है इसमें मिस्टेक में कैसे रन करते हैं चलते हैं हाँ एक बार करके देखिए मतलब अभी 2 दिन हैं तो चलता रहेगा वो चलता रहा है बट वो आ नहीं रहा है विजुअलाइजेशन मैं फिर ये हो गया ये मिस्टेक 2 के लिए हम लोग मिस्टेक का पहला वाला वर्जन यूज करना वो उसका सर्वर वेब सर्वर अवेलेबल है बट उसमें फिर सिर्फ एनआई ही होगा बट स्टिल हमें मिस्टेक का हमारा रिस्क में तो पता चल जाएगा ना चल आके शुरू में क्या करना बाकी सब तक हम फिगर आउट कर लेंगे बात करेंगे So we'll get escalated. Hands up.
मोबाइल में चले सब
हां साल खाना तो हम लगा था बस मेरा काम है अभी तो लगा नहीं काम बहुत ज्यादा
चलो टाइम क्या हो 1 घंटा यार लिखना
मैंने भी स्क्रीनशॉट लिया चाहिए तो चाहिए
हाँ एक 200 रजिस्टर किया था
आई 69 थे हाईएस्ट 69 किया
था 9 लोग तो अभी थे
कितने आते हैं हम पता है 9 लोग अपन थे मैंने भी अटेंड किया था मैंने रजिस्टर करके नहीं जाना था मुझे एक सर्टिफिकेट मिलेगा क्या
नहीं मिलेगा
नहीं मिलेगा मेरे उस पे लिखवा दो अंकिता वर्मा है
कहाँ चल रहा है ना
सब परिवार से
परिवार आता है आपको
मैं और अंकिता का साथ बैठा
फिर उसमें अगली बार इनविटेशन करेंगे कि हमारे अगले एमएलटीबी में से इनकी दीदी जरूर आए
आपको तो आना ही पड़ेगा
आपको आना पड़ेगा
यार आवाज नहीं आ रही है मैंने पूछा था पूरी तरह से आवाज नहीं आ रही है क्या मैंने करके इस लड़की की वजह से मैं सुन नहीं पा रही इसके दो दिमाग चल रहे थे एक तो मैम क्या कर रही है और एक इसमें क्या चल रहा है
कौन सा माइक
You are not multitasking. You तू सिर्फ मैम को ही देख रही थी और उन पे ही फोकस था तेरा मैं मैम का देख रही थी कि खाना खाना भैया मैं देख रही थी मैम का दिमाग कैसे चल रहा है चलो लेप खाली नहीं हुआ चित्र वाला ट्रायंगल शेप का बटन है चल जा रहे तू दिखा के नहीं यार मैं भी थक गई मैंने प्यार से बोला है
नहीं आएगा
इस इतने प्यार पे तो मर जाती है अभी तू मर जा लेट जाती हो मैं लेट जाती है लेकिन फ्लोर गंदा है
नहीं पीछे से जा रहा है
इसको बताओ मुझे
ये तो रखिए सॉरी वाली वो ज्यादा इंपॉर्टेंट
गले पे वो लग के
अब क्या करें उसी वक्त के लाइन करना यस एक व्यक्ति को वैसे वापस एडन के साथ रणजीत सिंह लक्ष्मी का यूएन वाले में चला गया इसमें गलती थी उसकी मैडम
The next generation of computation.
आ गया वन है ये इसमें कैसे आवाज है इसमें आवाज कैसे है अपलोड कर देते हैं हमने बताया आगे से मैं अच्छा एडिटिसी को भैया के दोस्त आए हाँ कौन से दोस्त आए वहाँ तो बैठे थे ये ना रिजल्ट मीटिंग करते की डेडलाइन एक्सटेंड होने वाली है कब तक? स्टार्ट करेंगे उसका पता है पैरामीटर भैया मना कर रहे हैं क्वेश्चन
MLTB and somehow with
Yeah, without further delay, I will start introducing about the RSG-India, what we do with RSG-India. So,
great professors and enthusiastic audience, we've been able to put this today. So without further ado, let's start. I would